So guess what? So far you've been doing really simple genetics problems, and now I'm going to make your life more complicated. What we have done so far has been complete dominance Punnett squares. All this means is that the dominant alleles are expressed over the recessive alleles. For example, if big R represents red flower allele and little r represents the white flower allele, then big R would give a phenotype of red, big R would give a phenotype of red, and little r would give a phenotype of white. You all know this already. If you were to do a Punnett square and you had a heterozygous organism, the gametes would be big R and little r. Same thing with the other one, big R and little r. You'd put it in a Punnett square like this. You'd cross mom's gametes with dad's gametes, and you would get this ratio. You would get 25% of the kids would most likely be big R, 50% of the kids would be big R, 25% of the kids will be little r, and if we were to look at the phenotype, we would see that 75% of the kids would be red and 25% of the kids would be white. There's nothing new here. This is what we have done over and over again. But guess what? This doesn't actually happen in nature very often. There are many other inheritance patterns that we are about to discuss. Sometimes alleles are not dominant or recessive. Instead, they are incomplete dominant. This means that the heterozygote organism is a blending of the two alleles. For instance, again, if big R represents the red flower allele and little r represents the white flower allele, big R would represent the phenotype of red. Here's where it's different. The heterozygous organism, big R, if it's incomplete dominant, it means it's a blending of red and white, which is pink. Little r is still white. So the only difference here is the heterozygote organism is a different color in this example. Let's do the same problem we did before. We have mom is heterozygous, here are her gametes. Dad is heterozygous, here are his gametes. Let's put it in a Punnett square like normal, and let's cross mom's egg with dad's sperm and see what we get. Again, we get the same genotype in the offspring. 25% have the genotype big R, 50% big R, 25% little r. But the phenotype is now different. 25% of the kids will have red flowers. 50% of the kids will be pink, and 25% will be white. If you are to ever get an incomplete dominant practice problem or test problem, it will make sure to say it in the paragraph. It will say, the way that flower color or the way that hair color is inherited is through incomplete dominant. That's a clue for you to know that a heterozygote organism, one that has a capital letter and a lowercase letter, have a blending of the colors or a blending of the heights or a blending of the alleles, whatever we're talking about. So notice the difference. In the previous one, 75% were red. In this one, 25% are red and 50% are pink. Codominance is when both alleles are expressed in a heterozygote. So we're no longer blending the alleles, we're expressing both. Again, the same example, big R would still have a phenotype of red. But the heterozygote, big R, would have red and white. Little r would still be the same, which is white. So let's review. Normal dominant recessive stuff, big R would be red. In incomplete dominance, big R would be pink. And in codominance, big R is red and white. Let's look at an example. Again, here's the parents that are heterozygous. They make the alleles big R in each gamete. Here's the dad. In each gamete, we have a big R and a little r. We put it in a Punnett square, and we cross them like normal. Look at the difference. Genotype still stays the same, 25% big R, 50% big R, 25% little r, but the phenotype is different. 25% are red, 50% are red and white, and 25% are white. You're not doing anything really different except you're reading the phenotypes different. So when you read a problem, make sure you're reading to find out if something is dominant or recessive, or it's incomplete dominant, or it's codominant. You have to read really closely or else you're going to get the phenotype wrong in the end. Okay, ready for it to get more complicated? So far, we've been talking about genes that have nothing to do with your sex. So far, it's just been about hair color or eye color, and it doesn't matter if you're a boy or a girl. Well, here's where it starts to matter. Remember, we have 2 of every chromosome, and therefore 2 of every gene or allele. We also have 2 chromosomes that determine our sex, the X and Y chromosome. If you look at the karyotype in the bottom right-hand corner here, you'll notice that the last 2 chromosomes are the X and the Y chromosome. So let's pretend you had a bunch of free time and you wanted to count all of these up. You would find that there are 46 chromosomes, 23 pairs of chromosomes. But notice down here, instead of numbers, it's labeled X and Y. You could either have 2 X chromosomes, and therefore you're a female, but if you inherit a Y from your dad and an X from your mom, then you're a boy. Notice something, that it's the father who actually determines the sex of the child. The mom is always going to give an X chromosome to their kid, but the father could either give an X chromosome and therefore make it a girl, or give it a Y chromosome and therefore make it a boy. Fathers are the ones in charge of determining if the child is a male or a female. Look at this baby's chromosomes. Again, you could count up all the chromosomes and you'd find that there are 46 chromosomes or 23 pairs of chromosomes, but look at the end. Is this child a boy or a girl? Looks like it's a girl. It has 2 X chromosomes. Let's figure out the probability of having a girl or a boy kid. First, we have to figure out the gametes. Males have 1 X and 1 Y in each of their cells, and therefore after meiosis, there would be an X in one cell and a Y in the other cell. Females have 2 X chromosomes, so after meiosis, there'd be an X in one gamete or egg and an X in the other gamete or egg. You then would cross them like normal, so you take the females and the males, put it together, and look what you have. The chance of you having a boy or a girl is 50-50, half. So really, every single time you potentially have sex and potentially would have a child, you would have a 50% chance of having a girl and a 50% chance of having a boy. Here's where it gets a little more complicated. Any genes that are found on the X chromosome are called sex-linked or X-linked genes. Remember how we have about 20,000 genes? Well, most of those 20,000 genes are found on the other 44 chromosomes. But the remaining 2 chromosomes, either 2 X's or an X and a Y, have some of the remaining genes. Therefore, they are called sex-linked or X-linked genes. One common example of sex-linked genes is hemophilia. Hemophilia is a blood disorder. If you have the allele for hemophilia, then you cannot properly clot your blood. And if you get a cut, it may continue to bleed and bleed and bleed and never stop, and you would potentially die. Let's look at the possible genotypes and phenotypes of people with hemophilia. Now be prepared, the way you write these sex-linked genes is a little complicated. Because hemophilia is found on the X chromosome, you have to write the X chromosome and potentially the Y chromosome when writing out genotypes. So in this little example, it says that hemophilia is sex-linked recessive. Therefore, the way that you would write this out is if you're writing a female, you would write XX because females have 2 X's. But if they are normal, meaning they don't have hemophilia, they would be capital H's because hemophilia is going to be represented as a recessive allele, so it would be a lowercase h. So a normal female would be X big H, X big H. This would also be a normal female. She's X big H, she has the normal allele, but she also has the hemophilia allele. Because big H is dominant to little h, this person is still normal. They do not have hemophilia. Although sometimes they're called a carrier because they are carrying the hemophilia gene or hemophilia allele, but they're not actually expressing it. This female has hemophilia. We know she's a female because there are 2 X's, and we also know she has hemophilia because she has the 2 recessive hemophilia alleles. This is a normal male. Remember, hemophilia is on the X chromosome, so you only have to write the letter H next to the X chromosome. It is not found on the Y chromosome, so males only have one shot here. They either have the good normal allele, or in this case, they have the recessive hemophilia allele. This male is normal because he has the normal dominant allele. This man has hemophilia because he has the hemophilia recessive allele on his X chromosome. The royal family in Britain actually has a high percentage of people with hemophilia. It all started with Queen Victoria up here. She was the carrier, or you could say she was heterozygous. She was X big H, X little h. And notice as you look through their family tree, there's actually a lot of people with hemophilia because she originally carried it, had some kids who married some other people who had hemophilia, and it got passed down to all their ancestors. The people that are alive today are right down here. See, William and Henry. They forgot to update Mary and Kate, and then they had a kid, I think George or something. So this is a little bit of an old pedigree, a little old family tree. Every year, people have a lot of trouble of setting up Punnett squares that are sex-linked, so pay attention. Most people want to set them up like we're doing a normal Punnett square, like I have right here. They would just do H's and that's it. But that's wrong. If you are to do a sex-linked Punnett square, you have to show the X's and Y's. This is a female. She is a carrier. This is a male. He is normal. The way that we do all Punnett squares is you first have to figure out the gametes. The female's gametes you separate like normal. These X's go to one egg, these X's go to the other egg. It just so happens that the hemophilia or normal allele are on the X chromosome, so they go together. Males do the same. Separate out the X, separate out the Y. Put them in a Punnett square. Cross the male sperm with the female egg, and here are all the possibilities. Then you can determine who has hemophilia and who doesn't. This female does not have hemophilia. This female does not have hemophilia, although she's considered a carrier because she's carrying the recessive hemophilia allele. This male does not have hemophilia because he has the normal allele. This male does have hemophilia because he has the recessive hemophilia allele. Another example of a sex-linked or X-linked trait is color blindness. Can anyone not see the number inside of this circle? Most of you should be able to see the green 7 inside of this circle to the right. If not, you may be colorblind. 10% of males are colorblind, so every year that means I have 1 or 2 students who cannot see the difference between greens and reds. Let's look at their genotypes. Color blindness is sex-linked recessive, just like hemophilia. So I'll represent it with a lowercase b. This is a normal female. She does not have any sex-linked recessive colorblind allele, so she's X big B, X big B. This is a woman who's also normal, but you'd call her a carrier because she carries the colorblind recessive allele on one of her X chromosomes. This is a colorblind female. She has both of the recessive colorblind alleles on each one of her X's. This is a normal male. He has a normal allele. This is a colorblind male. He has the colorblind recessive allele on his X chromosome. Again, sex-linked traits are more common in males because they only have one X chromosome. Females, on the other hand, have an opportunity to cover it up with a good dominant allele so you don't actually express the recessive disorder. Males, on the other hand, don't have a second X. So because they only have one X, it is way more common for men to have hemophilia, color blindness, muscular dystrophy, all the sex-linked gene disorders. Here's an example of a pedigree or a family tree that's showing the color blindness in this family. Notice the grandfather is colorblind. Down here, this woman is colorblind. Their son is colorblind. And in this case, the grandson is colorblind as well. This actually brings us nicely into discussing pedigrees, which is a way that you can trace traits throughout a family. The way you draw a pedigree is by using squares and circles. Circles represent the female in the family, and squares represent the males in the family. Whatever trait you're tracing in your family should be shaded. For example, if you want to see how common, let's say, rolling your tongue is in your family, you would shade the shapes of anyone in your family that could roll their tongue. Look at the picture up here. Let's pretend it's tongue rolling again. Looks like this woman can roll her tongue, this woman can roll her tongue, this woman, this man, and this man can roll their tongue. Let's try a few practice problems. This is a pretty simple one. So we have 2 parents have a kid. Question is, did the parents have a boy or a girl? Well, because it's a circle, that means they had a girl. Is the shaded trait a dominant or recessive trait? Here's where it gets a little harder. If it's a dominant trait, that means the parents must have had it because it would have to show up in the parents. So it is not a dominant trait because the parents don't have it, only the child does. If it's a recessive trait, The parents could be heterozygous, like they could be big A, little a, big A, little a, and the child could have the 25% chance of getting the little a from each parent, and therefore this could be a recessive trait because the kid has it, but the parents don't. What are the genotypes of the family members? Well, like I just said, the mom could be big A, little a, the dad could be big A, little a, and the kid, because it's a shaded trait and we've determined it's recessive, would have to be little a, little a. Let's try this one. Did the parents have a boy or a girl? Looks like it's a square, so it must be a boy. Is the trait dominant or recessive? Well, if the trait is recessive, this parent would be little a, this one would be little a, and the only possibility of the kid, if you were to do a Punnett square, would be little a. But the kid isn't shaded. The kid doesn't have the recessive disorder. Therefore, it is not a recessive trait. It must be a dominant trait. Therefore, if it's a dominant trait, the parents could be big A, little a, this one could be big A, little a, and the kid could be little a because it does not have the dominant trait. I know these are really tricky, but don't worry, we'll practice these in class. Finally, here's your last example of a pedigree. You should be able to look at the pedigree and tell me if they're male or females and if they have the trait, and possibly if the trait is a dominant trait or a recessive trait. Okay, so let's add another layer of complexity to some of these problems. Sometimes genes don't just have two alleles, like a big A and a little a. Sometimes genes have 3 or more alleles. This is called multiple alleles. Multiple means many, alleles means versions of a gene. So multiple alleles means instead of having 2 alleles or 2 versions of a gene, you actually have 3 or more alleles. An example of multiple alleles would be rabbit fur color. For instance, you could have a rabbit that has 2 big C's, it would be a dark gray rabbit. Or you could have a rabbit that's called a chinchilla rabbit, it's CCH, CCH. Or you could have a rabbit that's CHCH, that's a Himalayan rabbit. Or you could have a rabbit that's little c, that's a white rabbit. Notice there are 4 possible alleles. Instead of just being like big letter, lowercase letter, capital versus lowercase, instead there are multiple alleles, more than 2. Therefore, it's considered multiple alleles. An example of multiple alleles and codominance is blood types. There are 4 types of blood found in humans: O, A, B, and AB. The possible alleles that make up those blood types are IA, IB, and little I, and they can combine in a variety of different orders to create all of these blood types. Notice here, type A blood, the phenotype, has possible genotypes of IA or IA little I. B blood has a possible genotypes of IB or IB little I. AB has a possible genotypes of IB. This shows that it's codominant because both A and B are being expressed. O, on the other hand, has little I as its genotype. Don't have to memorize all this, but the point of me showing you this is that there are multiple alleles, more than just 2. Therefore, it's a type of multiple allele inheritance. Let's try a practice problem. A woman who is IA has a child with a man who is IB. What is the woman's phenotype? Well, if you look at the chart from before, IA means the phenotype is type A blood. The man's phenotype is type AB blood. That's an example of codominance. What is the probability their child will have type A blood? Well, the first thing we do is we figure out their gametes. If this is the woman, you separate out their alleles into separate gametes, and her gametes are IA. Put it in the Punnett square. Same thing with the man. Separate out his alleles. We have IA in one, IB in the other. Put it in the Punnett square. Cross the eggs with the sperm and look at the possible children. Here's the results. The probability that their kid will have type A blood, if you look at the 4 kids, is 2 out of 4, or half, 50%. Another type of inheritance is polygenic traits. It means multiple genes affect a trait. Poly means multiple, genic means genes. This means that there are a variety of different genes that will influence one type of characteristic. For instance, eye color, hair color, height, weight, intelligence, skin color. It's not just one set of genes that influences these traits, it's many types of genes. The most common example of polygenic traits, of which there are a lot in the human population, is skin color. There are actually 3 genes that determine your skin color: the A gene, the B gene, and the C gene. And the combination of all the A alleles, B alleles, and C alleles determine what your skin color is. Look around your neighborhood, look around the school. People aren't just either pale or dark. There's a wide range of skin colors, and that's because there are multiple genes that affect skin colors. It's a polygenic trait. Finally, I swear, the last layer of complexity. This is it. This is as hard as it gets. It will get no harder. Reminder that the law of segregation states that during meiosis, alleles separate from each other into separate gametes. You know this really well. If you start with little h, an allele goes into one gamete, an allele goes into the other gamete. Let's try this one, heterozygous. Big B, little b, separate into separate gametes. Same deal. You know this really well. In this particular example, let's say dad is homozygous dominant. That means each one of his gametes has one dominant allele. Mom is homozygous recessive, therefore in each of her gametes after meiosis, she has a recessive allele in each. Big H and little h come together through fertilization to create a zygote that's heterozygous. This is just one particular example. That's the law of segregation. The reason I bring this up is because not only can you cross one trait at a time, but you can cross 2 genes or 2 traits at a time. And we need to know the law of segregation plus this other law coming up in order to do them. For example, we can compare height and hair color. Let's pretend tall, big T, is dominant to short, which will be little t. Also, brown hair, which is big H, is dominant to blonde hair, little h. We can look at these both at the exact same time. Let's pretend a man is big T, big H, big H. What is his phenotype? So if you first look at the T's, it looks like he's tall, and then you look at the H's, it looks like he has brown hair. So his phenotype is tall with brown hair. A woman's genotype is big T, little h, little h. What's her phenotype? So let's look at the T's first. She's tall because she has one tall dominant allele, and she has blonde hair because she has 2 recessive blonde hair alleles. So she's tall with blonde hair. The reason why I'm bringing up these laws is because you need to understand the laws in order to do practice problems of those dihybrid crosses. If on different chromosomes, each pair of alleles separate independently of each other during meiosis. This is really important when crossing 2 or more traits at once, which are called dihybrid crosses. So look at this picture over here. Remember, homologous chromosomes separate independently of each other. They separate from each other during meiosis. It does not matter where the height allele separate, or the eye color allele separate, or the intelligence allele separates. It does not influence how the other ones separate into the gametes. For instance, let's pretend your mom's egg has the brown hair allele, but also has the freckle allele. It does not matter what alleles go into what gametes. They separate independently and randomly of each other. A good way to explain this is by looking at peas. Peas can either be yellow or green, or round or wrinkled. We'll have color represented as Y's and shape represented as R's. So there are possible ones. You could be yellow or round, you could be green and round, you could be yellow and wrinkled, or green and wrinkled. Now, if Y's represent color and R's represent shape, they could line up like this in meiosis. And if they line up like that in meiosis, they're going to separate the Y's and R's into individual gametes. The ones on the left go to the left, the ones on the right go to the right. But the Y's and R's could also align this way. And if that's the case, the Y and the R go here, and the Y and the R go there. The point of me showing you all of this is to show you it does not matter how, in this case, the Y's and the R's separate from each other in meiosis. You get a whole variety of different Y's and R's in each gamete. You could then take all of those gametes, so here are all the possible gametes, Y's and R's, and you could put it in a Punnett square. Look how big the Punnett square is. It's a 4 by 4. You could then cross all the mom's eggs with all the dad's sperms, and you could get 16 different possibilities in the kids. This is probably way more than you'll ever have to do on the MCAS, more than you'll have to do on my test, but I wanted to show you how this works in case there is a question. Again, if we are looking at two traits, let's pretend it's the Y trait and the R trait. They can separate independently into gametes. You put any combination of Y and R together in each gamete, you line them up on the Punnett square, and then you cross them together. Put the Y's together and put the R's together. Then you can look at the phenotype of each organism and determine that in this particular case, 9 out of 16 of these peas are going to be yellow and round, 3 out of 16 are going to be green and round, another 3 out of 16 are going to be yellow and wrinkled, and finally 1 out of 16 is going to be completely recessive, green and wrinkled. The only thing that I really want you to get out of this whole thing is that if you ever cross two heterozygous parents for both traits, so like A's are heterozygous and B's are heterozygous, or in this case up here, Y's and R's, the phenotypic ratio, what the offspring will always look like, will be 9, 3, 3, 1. It's a 9, 3, 3, 1 ratio. This is the question asked on the MCAS. If you cross two heterozygous organisms, the phenotype in the kids will be 9, 3, 3, 1. This is as hard as it possibly gets. Write down your questions and ask me in class.